Okay, I'm assuming we are live. Let me double check. Okay, yes. Okay, I'm assuming we are live. Good, sound is good as well. So let me turn this off. Perfect. Okay then, so this is now on the site also, I can see it. Great. I guess everything is set on YouTube and yes, on Zoom naturally. With that, we can get started for today's lecture, which is also our first lecture for this semester. So the course is PNS Genomics, or Project and Seminar Genomics. We actually have two courses with this direction, but we use the Genomics title to refer to both of these courses. So with that, let me get started. So I'll first start by introducing myself. I am Can. I am a senior PhD student in the SAFA research group, and I'm also a lecturer at ETH Zürich. My research interest usually lies in the intersection between bioinformatics and also computer architecture. With that, what I mean is I'm interested in designing algorithms as well as architectures for example, real-time genome analysis. I'm also interested in how we can do similarity search or sequence analysis in a large space of genomic data. And also, of course, how can we co-design the algorithm and the hardware together? It's not just you implement the algorithm onto an existing hardware, but you rather try to think about what should change in the algorithm and also what should change in the hardware, so that these two can be orchestrated nicely together to accelerate certain tasks in genome analysis. I'm also very interested in genome editing. How do we, let's say, design algorithms and pipelines to perform genome editing experiments such that your editing, your genome editing will be successful and very precise at the end. And also, I'm interested in correcting errors in the sequencing data. So you can get to know our group and our research using these links. These links are available on YouTube in the video description. Actually, the links to the slides are available, and through that you can find all these information about these links and all the other information essentially. You can contact me using my Gmail address, and or you can essentially take a look at the papers and the other works, other talks that I'm delivering using this website. And also I'm posting some of these news, scientific related news, also on Twitter, or now it is referred as X, I guess. And in fact, I posted this very first lecture on X just a few minutes ago. So yeah, so what is the agenda for today? We will first cover the goal of this course, what will be the content of the weekly lectures, what you should expect from this course, as well as what we expect from students. We will cover the course logistics and also how you should be communicating with your mentors, which will be assigned to you. And we will have a very brief introduction to genome analysis, which actually we will start covering in depth starting from next week. So what are the key goals of this course? We have two main goals. First, we hope that you will, by the end of this course, you will understand the computational steps and also the challenges in genome analysis. Second, then hopefully you'll learn how to overcome these challenges with maybe just software, or perhaps hardware, software, and algorithm co-design. So to this end, we have 2 courses that we're offering each semester. One of these courses are called Project and Seminar, or PNS, Mobile Genomics. Here, a specific goal would be designing hardware and software mainly for constrained computational resources, for example, some low power designs. And the second course is called PNS Accelerating Genomics, which focuses on co-designing hardware and software to accelerate genome analysis. But in fact, we actually have sort of like a combined syllabus for both of these courses. As you may notice, and also on the course website, we will be delivering the lectures that will cover both of these courses, so that regardless if you are taking a mobile genomics course or accelerating genomics course, you will be watching the same lectures eventually. And essentially both of these projects, these courses are project-based courses. Eventually, by working on a project, you will understand and improve the cutting-edge techniques. You'll have a hands-on research exploration and experience with mentors. We will expect you to watch optional weekly lectures on genomics because it will help you understand what are the main steps in genome analysis and what the challenges are, so that you can get a grasp on and also a nice understanding on what we're trying to achieve with these projects. And also, of course, especially towards the end of the course, you will hopefully improve your research skills, especially if you want to be an independent researcher. You'll see how we approach the scientific problems. What are the important aspects to approach when approaching a scientific problem, and how can we essentially solve these problems? And also then you will present your project to your peers and also to the mentors at the end of the semester. This is usually either right before the exam start in January, or somewhere basically sometime in February or later January. So we will communicate this with you basically as we approach to the end of the course. So let's see. Okay, yes. I don't see any issue with my audio. I'm just sometimes checking if there are any issues, just in case. Okay. All right. So what are these hands-on projects, right? So you already received a form. If you are taking this course, you already received an email from me, which is essentially a post from Moodle. And again, as a reminder, I suggest you to follow the Moodle posts very closely, and also even subscribe to these posts so that you can get the email also whenever we post something on Moodle. That is very important because we will announce important updates related to this course. For example, the form that you just received, right? The deadline about it, as well as perhaps like when we're going to ask you when we should be scheduling the project presentations and so on, so that we will be asking for your input and other things. But then eventually, when you indicate your skills and your preferences on the form, we will look at those, and then we will also look at the available projects we have, and then we will try to assign you to a project. So it may so happen that you may work with, again, another student on the same project, or maybe you may just solely be working on a project with a particular mentor. But essentially, what you're going to be doing, you will experimentally evaluate different heuristic algorithms, perhaps, and maybe architecture to observe their effect on end results. We will have optional weekly lectures, as I said, which will cover the basics of genome analysis to understand the computational steps of the entire genome analysis pipeline. But also towards the end of the course, we will also have these specialized lectures that will cover some of the papers basically that we're publishing in our group, so that you can see what are the challenges in the genome analysis steps, and actually how we are solving these challenges in our papers. So as I said, hopefully you will improve your basic knowledge in genome analysis, also technical skills in genome analysis and computer architecture, also critical thinking and analysis. You'll get some familiarity with key research directions, and then you'll also improve your technical presentation skills of your project by the end of this course. So what are the prerequisites of this course? First and foremost, since we're getting this question a lot from the students, you don't have to have prior knowledge in bioinformatics or genome analysis. Hopefully, you'll gain this knowledge throughout these optional weekly lectures we will be releasing, as well as the papers or the materials that your mentors will be sending you as you meet with your mentors. So whatever the knowledge is needed to complete or to have progress on your projects, you will gain them eventually throughout this course. But of course, as with anything else, everything right in life, we expect you to have an interest in whatever you're doing, right? In this course, this means that we need an interest from you in optimizing and efficiency and solving complex problems, right? So it's really good that if you approach the problems that you're trying to solve with an interest, so that because then you will essentially experience some obstacles, right? You will face with challenges. And if you don't have interest in solving these challenges, then you won't be able to have good progress. But when you have an interest, you'll talk to your mentor, you'll basically look at other materials and so on, and eventually you'll make a good progress in this project. And who knows, maybe you will want to continue in this field, which is really something really nice to hear from the students taking this course in the past. It is really good if you have some basic or maybe even good knowledge in C or C++ programming language, because in most projects, regardless if it is a hardware focused or algorithm focused project, you'll need to change some implementation or implement something from scratch. And we will mostly be doing these using C or C++. It is very important if you have some experience in bash scripting. This is important because most of you will gain access to our servers, and then you will be running perhaps some tools or your implementation on the server. But in order for you to do that, you'll need to know, for example, how to SSH, how to log into our servers, and also how to use the server, right? So we will be sending you some introductory material for this, but if you already have this knowledge beforehand, then this means that you'll save a lot of time getting used to these materials and how to use the server. It is not required, but if you already have taken, for example, some previous coursework such as digital design and computer architecture, or an equivalent course where you get an understanding of how computers work, right, in lower level, and how we can design basically or architect a computer from scratch. It is really important so that you can gain bring some insights already to this course. Again, this is not required, but if you already have an experience in either FPGA or GPU programming, it is really beneficial. And especially if you have such a knowledge, please indicate this in your form so that we can assign the projects accordingly to you. So the requirements about this course, what we expect from you so that you can make a good progress. One is, although this is optional, we expect you to watch the weekly lectures. The schedule is already on the course website. You'll get the links to the PDFs and the PowerPoint, as well as the YouTube links right on the course website. So you can just check the course website and then see when we're having the next lecture, and then perhaps watch it live or maybe later if you're not available at that particular time. You'll have projects that you're going to be working on. In order for you to pass this course, it is required that you have a progress on this project, which means that you'll build, implement, and design basically something to solve a problem that your mentor will be describing to you. So this means that you'll have under close mentor guidance. There will be a mentor assigned to you, and then you will be working with the mentor. We expect that around 6 hours of work that you'll put on this course per week. Of course, this can change time to time. You may have exams and other things, but this is more or less what we expect from you every week. And of course, it is very important that you have regular meetings with your mentors. With that said, communication with your mentors is very important. Please regularly check the course website, Moodle. Our instant communication channel is Discord. If you already have a Discord handle, a username, please let your mentor know, or you can use emails to communicate, but Discord is a bit more user-friendly. So timely responses to your mentors is required and is really needed to have a progress on your projects. And at the end of the course, when you have progress on your project, you're going to present that progress to your peers. So there is a mistake here. So this is the last week, perhaps, of December or January. And also you'll be uploading all the materials, for example, code and results that you have, that you already essentially implemented throughout the course to a Git repository. If you have a project with significant progress and good results, we will support you for publication. So how do we assign the projects? And right, so you'll provide your interest, and then we will look at your interests and skills, and then we will assign the projects to you. And the mentor will contact you accordingly. You'll meet with your mentors regularly to show your progress. So with that said, who are the mentors? We have a bunch of mentors, including myself. I already introduced myself. We also have Konstantina, who is the senior researcher in our group. She's essentially experienced in, of course, in bioinformatics, but also hardware software co-design for genome analysis. We also have Nika, who is a PhD student in our group. She's very interested in emerging technologies, for example, emerging memory technologies, and as well as other technologies such as doing computation within the storage. And she's basically bringing that perspective to genome analysis and asking the question, how can we do genome analysis perhaps within the storage or using these emerging technologies? We also have Clea. She's visiting us from Bilkent University. She's supervised by Can Alkan at Bilkent. She's mainly interested in designing algorithms, specifically perhaps for, let's say, specifically or accurately identifying copy number variations within a genome, which is very important, especially if you are interested in recognizing whether someone has cancer or not, right? And then she's also interested in designing algorithms for assembly and so on. But also she's interested in the hardware side of bioinformatics and as well as the processing in memory. We also have Zülal. Zülal is a PhD student at Bilkent University, and she's also supervised by Can Alkan. Similar to Clea, she's again interested in designing algorithms for bioinformatics and computational biology, but as well as in designing, implementing basically these bioinformatics and genome analysis workloads for particular hardware such as FPGA and GPU and so on. We also have Tolu. Tolu is he's been working with us to complete his bachelor's thesis. which he just did, and he's still working with us, and basically he's very interested in a similar direction what Nika is working on, as Nika was supervising him. Essentially, he is focusing on how to accelerate genome analysis, specifically in storage. But his interest is also very broad. He's exploring basically many fields in genomics and also other directions. So this is a brief intro to the mentors. So this is a screenshot from our course website, and you can see the tentative plan of this course. So this can change, but this is how it looks like so far. This week we are basically talking about the logistics, but like a brief intro to the genomics as well towards the end of this lecture. But starting from next week, we will have a more have a deep dive into genome analysis. We will give you a broader overview of the steps in genome analysis, and in the upcoming weeks we will show you how you get the data, how you get the genomic data, and how you analyze it using particular steps and so on. And then towards the end of the course, we will talk about the papers that we already published in our group. We've been giving this lecture for many semesters, so this is these are the links from the previous semester. You can already take a look at the slides and the lectures that we covered last semester using these links, and also we have a full playlist also to the lectures from the previous semester. So you can reach all these links using the as this slide is available again, as I said, in the course, in the video description on the YouTube and also on the course website. And also these are all the links to the previous semesters. So with that, let's very quickly cover what we're doing essentially with this course with a very brief introduction. We essentially aim for fast, scalable, and accurate genome analysis. And there are very good reasons for that, because the genome analysis is important for many reasons. For example, it helps us understand the genetic variations between individuals, right? If a particular population shows basically statistically important phenotype, right? For example, they are prone to a particular disease because of a certain variation that they have in their genome compared to all other population in the world, right? So we can study this. We can say, oh, when this population has this particular variation in their genome, it is possible that they're going to be prone to a particular disease. And this is very important to understand so that we can link the diseases to the variations in the genome. And so that we can even try to develop solutions for these diseases, right? Maybe even edit that particular region in the genome so that you can sort of disable that functionality so that that particular population won't have that disease anymore. So you can already see how important this is to understand genetic variations, but in general, how important it is to analyze the genomics. But it's not limited to that. We want to understand the species. We want to compare the species to each other to understand maybe our common ancestors, right? We want to understand the evolution between organisms. This is very important, especially in the case of COVID, right? So literally, scientists can see how COVID is evolving so that we can already be ahead of it and then see whether it will evolve into something dangerous or not, and so that we can start designing drugs accordingly. With that said, genome analysis is also important for predicting the presence and relative abundance of microbes, for example, or pathogens in a sample. We can take a sample from, for example, a New York metro station and then see whether a very harmful pathogen is growing in that sample so that we can take early action based on this analysis. Again, with that said, this becomes very important for the rapid surveillance of disease outbreaks. We already had a very recent COVID outbreak, so this means that if we could have a rapid surveillance everywhere on Earth, and then analyze it very quickly, we could perhaps see it very early that something harmful going on, and so that we can take a very early action based on that. And this is also very important for personalized medicine, as I already said. Maybe we can design drugs, right, that is individually designed for you, that is individually prescribed to you based on what you have in your genome, right? Because we can understand how that drug will have an effect, basically, on your body, or what sort of phenotype it will, what sort of effects it will generate when you take that drug. So you can already see essentially why this is important to have an analysis of genome in a very accurate way, but also in a very fast and scalable way, because it's also about understanding harmful samples in a very quick way in the population. So how do we do it? How do we analyze the genome? We usually start with a sample collection. For example, assume we take a swab from your mouth, or we take a blood from your tissue, and then we collect that sample. Then what we do is, like, there are a bunch of steps that essentially chops this DNA into multiple fragments, perhaps some adapters, or some, let's say, specialized, let's say, tools that are attached to the ends of the DNA or RNA, and perhaps maybe if we have a low amount of DNA from your sample, we can even amplify it, we can copy it, duplicate it in many ways, and so on. So this is essentially called library preparation. We prepare the library, we prepare your sample in a good way so that it can talk to a machine that we call a sequencing machine. Right. So once we prepare this library according to the manual, perhaps, that the sequencing device is giving you, then we input that library to the sequencing machine, and then sequencing machine runs in some way that we will also explain in the coming lectures, that such that it generates some data that we call raw sequencing data. It could be sets of images, it could be electrical signal, which reflects basically the individual bases, the individual nucleotides that you have in your genome. Then this raw sequencing data is analyzed in some way, using some steps, so that we can gain insights from your DNA. So we are mainly interested in the computational steps that we perform here, including raw sequencing data, as well as the next steps we perform to analyze your genome. So there are many different sequencing devices. All of them have different requirements, different outputs, and so on. We will cover all of these, but we are mainly very interested in basically looking at newer sequencing technologies and then understanding what we can do with them. We will cover, again, many works related to this direction. In my opinion, they are very exciting. We also look at essentially what can be enabled in the future by looking at these newer sequencing devices. For example, can we do sequencing and genome analysis in field in a portable way? Assume you plug the sequencer to your mobile phone, or maybe to a drone, and then you go to a field, and then you analyze an organism, right? This can be anything. And doing this in a very efficient and quick way, and again, in a power efficient way, is very important. So then what is an intelligent genome analysis? It means many things. We want to analyze the genome in a fast way, in a large scale way, in an accurate way, maybe using intelligent architectures, and also maybe protecting the privacy. And all of this mean essentially we want to use intelligent architectures to analyze the genome by looking at the requirements or the characteristics of the data that is coming out from this device that we call sequencing machine, and then asking the question what sort of device we should use. And this could even be a mobile device, right? And then to achieve this, we are looking at the entire stack of computer architecture, right? So starting from the problem almost down to the electrons level, but essentially we're usually stopping at the devices level, and then we're asking the question, how should we co-design this entire stack so that the analysis becomes accurate but also fast? And what we really observe is that the data generation is really not a problem, right? So these devices the sequencing devices are really specialized at generating the data at a very, very fast rate. But then what the problem is, we are trying to analyze this data using a general purpose machine. This general purpose machine could be your personal computer, or it could be a server sitting at the cloud, but they don't usually have an idea of the characteristics of this data, what the requirements what are the requirements of this data, right? Knowing this actually could give you an edge, so that you can perhaps design the hardware accordingly, so that you're not bottlenecked by data analysis. So this is very important, and then we're going to be covering also in the upcoming lectures. And these are also the steps in genome analysis, main steps in genome analysis that we are going to be covering. So as I said, the genome analysis usually starts by the data generation, right? How do we generate the data? So you input the DNA molecule, you get some raw data, and then how do you analyze this data, right? There are some steps to it. And then once you process that raw data, so what are the next steps? This is a bit also related about the limitations and the challenges in genome analysis, or the sequencing data that we're getting from these devices. But then later on, once you analyze it, how do you generate insights from the analyzed data, right? So we're going to be covering all of these steps in the upcoming lecture, so that you have an idea of essentially how we start from the very beginning and up until the end to get some important answers from your genome. But if you're already interested in looking at these steps, you can take a look at this paper that covers, again, almost all the steps from the beginning to the very end. These are also some recommended readings if you're interested in basically core algorithms in the sequence analysis and the genomics that we use even today, right? You can take a look at these textbooks. I'll also show you some materials from our group, which I believe provide a good overview also of the challenges in genome analysis, and also the core steps in genome analysis. For example, this work covers what are basically the next steps to accelerate genome analysis using perhaps a processing in memory, which you can do the computation either near or inside the memory devices. Again, a similar work, how you use FPGAs and also with a near memory, let's say, devices attached to it to accelerate important applications, not just genomics. This I already showed you that, like, the steps in genome analysis. And also this work covers a broad overview of the efforts in the field to accelerate genome analysis, what sort of steps we have in genome analysis, and what sort of acceleration efforts that we put to it to accelerate it. This is one work that shows how we essentially can accelerate important steps in genome analysis using processing in memory. We also organize workshops. We invite people from the field. For example, we invited people from NVIDIA, IBM, and also from institutes and universities from Michigan, for example, or in other places. Basically, we invite essentially people who are working in this direction, so that they give talks and they describe their takes, essentially their perspective on this direction, and so on. So this is a workshop that we organize jointly with the Recomp conference, which is one of the top conferences in the bioinformatics field. And then the entire workshop is available on YouTube, so you can take a look at this link and then see what sort of talks we had there. We also give talks to basically motivate or to describe what are the challenges in genome analysis, so you can take a look at all these talks to learn more about the steps in genome analysis. So these are all basically materials that you can reach using these slides. Yes, so I'm just flashing these slides just to show that we have many talks that we're also giving, and all of them are available, again, using these slides, but also using either my website or the Sapphire website. You can reach all these links to those talks. So with that, I will finish today's lecture. I don't see any questions on YouTube. I'll just wait for a few seconds just in case there are like some last minute questions. If not, we can end the meeting for today's lecture. Okay, so I don't see any other question that is coming up in the last minute. So with that, then thanks everyone for your attention. If you have any questions, feel free to send me an email. And I hope to see you next week in the next lecture.